Okay. Huh? Okay. So we start.
Hello everyone and welcome to today's session. Thank you for joining us for today's webinar. Good afternoon all. A warm welcome to all the participants of today's Hello everyone and welcome to today's session. Today's orientation session unlocking innovation through quality by design, a systematic approach to today's orientation session unlocking innovation approach to pharmaceutical drug development. KMB College of Pharmacy, Department of Pharmaceutical Chemistry, and Institutions Innovation Council for KMB College of Pharmacy, Perumbavoor.
Hello, good afternoon, all. Distinguished
You can
ladies
enter
and
your queries and comments in the chat box throughout the presentation. Thank you and have a happy learning. Now I request Dr. Harish Babu E. Professor, KMB College of Pharmacy to deliver the welcome address. Sir.
thank you, madam. Distinguished delegates, faculty members, students, and my dear friends, a warm good afternoon to one and all. Today we are conducting our orientation session on unblocking innovation through quality by design, a systematic approach to pharmaceutical drug development. I, Dr. Harish Babu, Professor KMP College of Pharmacy, extend warm wishes on behalf of KMP College of Pharmacy, Perumbavoor, which is run by Indira Gandhi Memorial Trust, Nellipuri, Kodamangala. I take this opportunity to convey my heartfelt gratitude to our management for giving continuous support. It's my immense pleasure to welcome our chairman, Haji Sri K. Pareed, who is always with us for any type of activities. On behalf of our Indira Gandhi family, I welcome you, sir. I cordially welcome the chief speaker for today's orientation session, Ms. Uday Raj, Consultant, Quality Assurance Audit Compliance, Solara Active Pharma Science Private Limited, Chennai. Welcome, madam. I'm so happy to welcome the leading power behind the success of our programs. The leading power and constant teamwork makes a program grand success. It's none other than our principal, Dr. J. Nanda Kumar. With most respect, I welcome you, sir. I welcome our faculty members of KMB College of Pharmacy, without whom this webinar won't be possible. I welcome you all. Any program success is its participants. It's my privilege to welcome you all participants join here. Welcome all. By seeking worship from God Almighty, let me conclude. Once again, welcome all to this orientation program of KMB College of Pharmacy. Hope you will find it very useful. Thank you.
A matter of great pleasure and honor for me. Welcome to our principal, Dr. J. Nandakumar sir. He is well known for his work on the field of education and experience in teaching for 19 years. Now, our principal sir, to today's resource person, please sir.
Thank you, madam. Thank you so much. So, a very warm good afternoon to everyone gathered here. It's my immense pleasure and privilege to welcome all of you for this fourth National Webinar Cum Orientation Series on behalf of KMP College of Pharmacy, Perumbavoor, Kerala, and on my own behalf. I would like to introduce our today's speaker, Mrs. Udaya Raj. She is working as a consultant in Quality Assurance Department from Solara Active Pharma Science Private Limited, Chennai, Tamil Nadu. Mrs. Udaya Raj graduated from the Tamil Nadu Dr. MGR Medical University, Chennai in the year of 2001 and post-graduated from the same university in the year 2015. She is a certified training provider from Varsigma, New Delhi. She is a certified training provider from Varsigma, New Delhi. She started her career as a quality assurance and quality control chemist at Madras Research and Analytical Laboratory, Chennai, during the period of September 2001 to May 2003. Later, she had joined as senior research scientist at Twist Life Sciences, Tambaram, Chennai, during the period of May 2003 to July 2006. Later, she contributed her professional service as team leader in analytical research and development at Archit Healthcare Limited, Irungattukottai, Chennai, Tamil Nadu. She is having sound knowledge in root cause analysis, implementation of CABA, change control, out of specifications, control procedure and handling of ICH, GLP, GMP and GCP and regulatory requirements also. In addition to that, she is having versatile experience in calibration and handling of all kinds of analytical equipments, especially critical analytical equipment, and familiar with guidelines of various regulatory agencies like US FDA, ICH, WHO, ANVISA, MVA, OECD, GLP, etc. Presently, she is continuing her service as a consultant in Quality Assurance Department, Solara Active Pharma Sciences Private Limited, Chennai, Tamil Nadu. Hope this session will be useful to everyone. Now, with this small introductory note, I shall hand over the session to Mrs. Udaya Raj. Please, madam.
Thank you so much, sir. Thanks for your warm welcome. Hope I'm audible to all. Can somebody give me a reply? Am I audible? Hello?
Yes, ma'am, we can hear you.
Hello?
Audible, madam. No problem.
Yes,
Okay.
ma'am.
Yeah, fine. Thank you. There is a little bit of internet connectivity issues here. I thought it's not. Okay, fine. Thank you. So prior to that, in life, we want everything in quality product. We want a quality phone. We want to spend a quality time with our beloved ones. Even our lifestyle should be a quality one. That much quality in our life. When quality cannot be separated, like can neither be separated from a drug product. Every pharmaceutical company knows the pain of bringing out a quality product. In this pandemic situation, we also expecting a product, a vaccine, which will cure. So many of us had a question: why manufacture a vaccine? What is there? Why it is so much taking so much of time? The question and the answer in this session. Welcome to the webinar. Let me my screen. Give me a moment. I'm just sharing. Please let me know if my screen is visible. Hello. Hello.
Yes, ma'am, your screen is not shared.
Am I audible? Is my screen visible for everyone?
No, ma'am, screen is not visible. You are audible.
No, I couldn't hear.
Not visible, ma'am.
Now is it okay?
No, ma'am.
Give me a moment, maybe. Just click onto the share screen. Okay. Hello. Am I audible, sir, now?
Yes, ma'am, audible.
Ma'am, can you please switch off your video and then try, ma'am? Can you please switch off your video and then try?
I'm just trying to present my screen. Okay, that's also a good idea. If my screen is visible, please let me know so that I can able to present.
No, ma'am. It's not visible.
Sorry, not visible, right? Okay, 1 minute.
Madam,
So
not
I'm
visible,
just
madam.
trying
What
to,
happened, madam?
enable that screen. Yeah, sir.
Yeah, tell me.
Sir, shall I send the PPT to you right now so that you can present and I'll be explaining it to you or the IT team?
IT team, is it possible? Eldos, Sibi?
Because I think you can hear my voice.
Okay, please forward to principal@kmpcp.org.
Ma'am, can you send your PPT to events at kmpcp.org?
Yeah, sure. Just a moment. Give me a moment. Just forwarding right now.
Ma'am, you can check in message, chat box.
Yeah.
I have shared you the mail ID.
Yes. I'll just share right now. Ma'am, can you please check now?
Okay, ma'am. Ma'am, we haven't received your slide, ma'am.
Just a moment. Maybe it is taking a bit of a time.
Yeah.
Events.
Events at kmbcp.org.
Yeah, done it.
Yeah, let me check, ma'am.
Yeah. Thank you.
Yes, ma'am, we received. We are presenting.
Yeah, okay.
Ma'am?
Yes, thank you. Yes.
Yes, ma'am, you may start.
Yeah, thank you so much. And apologies for the technical glitch. And can you just
It's okay.
maximize it, put it in a presentation mode so that everyone can see very clearly? And is that been done? Hello.
Madam, it's audible, madam. No problem. Carry on.
Yeah, no, I couldn't able to see the screen. The screen is now not visible for me. That's what. Okay, I'll do one thing. I'll just see my PPT and present so that you can present the for the other people. So I'll just do it if the voice is very clear. So without any delay, let me start this presentation. So the point here is unlocking innovation through quality by design. At the end of the presentation, you will understand what is QBD, what are all the elements of QBD, what is called critical quality attribute, and what are all the pharmaceutical quality system, what is the actual expectations, risk management, and of course, what is DOE, everything you will understand. So prior to that, we need to understand what are all the different stages in the drug development. Slide number 3. So the goal of a product development altogether, it is to bring out a quality product. That obviously it has to be it has to be safe for the patients, and it should be efficient too. So without that, we cannot build a quality product. Next slide, please. So if we see the different stages of drug development, we need to understand first. So before getting into the systemic approach of a product development, we should understand what are all the different steps in the drug development. Then only we can understand what is the systematic approach or what are all the proactive approach we have to go to into that mode. So first basic thing to drug development, to develop any product, it is the first step is the literature review. We need to build a strategy for the formulation first. So parallelly, the analytical method development will be go on. So during the lab scale batches and the analytical method development, first thing is you should analyze about your product. When the product is given to you, you have to see what is that molecular formula, what is the atomic weight, what is its different chemical properties. Without knowing that, you cannot solubilize a drug. So first, it's very essential that you should understand the formulation and then move on to the analytical method development. If you know the solubility of a product, obviously you can prepare a solution and you can inject into the system. That's what analytical method development is. So further, there is a development, there is a validation, there is a process validation. Those steps will go on. But in a very concise manner, we have given over here. If you're going to develop a product, obviously you need a standard product, correct? So if you need a standard product, that is called as a reference product. When you research, when you market analysis, there is a lot of product like this is available. Hello? Am I audible?
Yes, ma'am. Please continue.
Yeah.
Yes, ma'am.
Audible, madam.
So there are a lot of products. Yeah, thank you. So there are a lot of products available. So you have to compare your product. Are you going to make the same kind of product or not? If you are going for same kind of product, then what are all the resources you need? What are all the input materials you need? That analysis you have to do at the beginning of any product development. Okay. So it is not just the formulation. Again, if you're coming back, the input material, we call that it is the API, active pharmaceutical ingredient, and the excipients. These two only will constitute the product. Okay. So these two products, you need to characterize like what kind of API you are going to choose as a key starting material for a product. Now, quality product cannot be built at the end. If you want a quality product at the end, the product which you are making or the product which you are using at the beginning should be a quality one. The point here is the API or excipients. Whichever you use, it should be of good quality. Then only you will get a good quality product. Okay, now you have selected your AP and expients. What is the next step? The next step is your formula optimization. How much amount of API you are going to take, how much amount of excipients you are going to take, what is the proportions, in which stages, how many stages you need to bring out that product. All these optimization you have to do, not just the mixing, grinding and all. It is the equipments which you are going to use in formulation as well as in analytical. So whenever you are going through all these steps, there are two wings, okay, two verticals you have to think about it. Only is the formulation strategy and another one is the analytical strategy. So the process which you are going through formulation, the same process you have to adapt in the analytical. Only thing is this is process and this is called as method. That's it. The equipment whichever you are going to select in the formulation, parallelly you have to decide which equipment you are going to analyze your product. Fine. The next step is you have analyzed, you have selected everything. You have selected your machine, you have selected your input materials, you have selected which method you need to adapt. Now it is the time for analyzing the substance. You are analyzing it, you are getting a result, and definitely it is not that the product which you have developed is of good quality, but at the same time it should be stable product. If you want to give an expiry date, it is must that you should use this stability study. Now comes the process optimization. Once you fix your stability, then you need to create your own specification. That's what in the second study it is written, process optimization specs. It specs nothing but product specification and the validated methods. Now you have developed your product, you have analyzed, you have given the expiry date for your product. The next step is to conduct a pilot and pivotal biostudy. Biostudy is nothing but clinical trials. Without that, you cannot bring out your product into the market. So before even bringing into the market, you need to file your product, and your product has to be approved by a regulatory agency called FDA to market in US. Or if you are going to market in some other region, then it is called as some other regulatory agency. In domestic, it is definitely there is an Indian FDA is also available, CDSCO, and you have to get the product approval from that particular regulatory agency to make your product development. Next slide, please. Now, the goal of a product development. So what is our goal? Our ultimate goal is the quality product, patient safety, efficacy. We have already seen it. And before even getting into the quality by design concept, we should know what is a pharmaceutical quality system is all about. Pharmaceutical quality system, in one shot, there is a 5 pillars which is very essential to build a quality management system in a company, in an organization. First is the production system, where you manufacture the product. The process control system, how do you optimize your product characteristics? That is called process control. Then comes your facilities and engineering. It is not just about the product and the process and the analytical method which defines the quality of a product. Of course, it is a facility too. What I'm saying is the biggest challenge in today's pharmaceutical industry is the contamination. Cross-contamination. The biggest contamination comes through the people. So the design of a facility has to be designed in such a way that when people enter into the premises, they have to be given some sort of training that is called good personal hygiene practices. What sort of governing they have to wear, what sort of chappals they have to wear, primary clothes, of course, it's not entered. There should be a changeover room should be there to enter into the classified area. So, of course, the entire facility should be easily cleanable to avoid all these contaminations. So that's why facility is also playing a major role in terms of a quality system. Then comes your laboratory system. Laboratory system, Without analysis, there is nothing. Whatever product you manufacture, you develop, it has to be analyzed by a validated method. So there should be a proper laboratory system. Of course, it should be separated out from the manufacturing area. Then comes your packing and labeling. There should be a proper packing and labeling system should be there. That area also should be near to the manufacturing area. Each and every label which is getting manufactured or which is getting used for that particular product has to be controlled manner. The person, whoever is involving in the packing activity, should get trained how to control this. So these are all the 5 pillars which actually build a quality system. Next. Now here comes what is quality by design. It's nothing but a systematic way, systematic, holistic and proactive approach to any pharmaceutical development. There was a time, whenever they say quality assurance department or quality control department, it is nothing but people, when they manufacture some products, they will come with the trials. Every time they'll go for some assumption, they'll come up with samples and they will hand it over to the quality control department and ask us to analyze and give the results. The quality control's responsibility is to give the results. So based on the results, again the formulation scientist will go, again he do some trials and then again he will come back. This is how the traditional way of development was going on. But at times, it was realized that for development itself, it is taking much of time, which in terms is giving a lot of pressure on the formulation scientists to bring out a product, to bring out a quality product to the market. It's taking more than 2 years, 2.5 years. Sometimes the product even will drop down because of there is no market for that product, and some other company would launch prior to the one particular company which is ready to launch. So there were challenges. So this concept has been brought into picture. So it is a systematic, holistic and proactive approach to any pharmaceutical development. It begins with the predefined objectives. There is no trial and error method. As on days, there is no trial and error method. Everything, every objective is well defined. So if you see the right hand side, you see this product knowledge, product specification, product performance, everything is designed. How the product has to behave, that is defined in the specification. So it should be a predefined objective. And this QBD concept, it emphasizes the product and process and understanding of process control. Whomsoever is developing the product, the particular scientist has to know about how the product will behave, how the process will come out, and how he has to control that process. He should have a broad knowledge, wide knowledge on the process. The person will be trained accordingly. Then only he will be ready to hand over that particular project. Now, based on this sound science and quality risk management, we'll see about this quality risk management at the end so that you will understand a bit afterwards. Now, I say also you will not understand what is the quality risk management all about. So the objective, final thing is like there should be pre-objective. It is well defined. This is how you have to move for your development process. Next. And many will be asking this question, why QBD? When I get in trial and error method, of course, I know how to do that. Why this QBD? Of course, this QBD ensures higher level of assurance of product quality, which you will not get it in trial and error method, and improved process and product understanding. Where it is going wrong, where I need to correct the process parameters, everything you will come to understand only with this approach. And monitoring, tracking, trending of product and process is very much possible with this QBD. But because most of the people who do trial and error, they just go and inject, prepare a sequence, they just inject into the system and see the results. There will not be any proper documentation. If the second scientist comes and checks, he will not be having any clue how this method has been developed, how this process has been developed. But in QBD, it is not the case. Everything you can track right from the beginning, what input material has been given and why the results has been deviated. So all these informations, you will get it in this approach. And of course, increase the efficiency of manufacturing process. By doing this, if you get a product efficacy by means of 90%, you definitely will get 99% of potency when you understand this concept. The objective is, by doing so, there is nothing called product failure. Your product will never fail if you follow this QBD approach. When there is a fixed approach, when there is a fixed parameter, you will do a lot of research on all other parameters and come out with solution. Yes, my product is going to yield this much of amount, this much accurate, and this will be having this much amount of impurities, and everything is well within the limits. There is no product failure. It actually will minimize all those product failures which has happened in the past. This, in fact, in turn, will save the cost of the company. Next slide, please. So, by seeing all these things, I want to give an overview of this QBD, what it is. There is something called quality target product profile. This is all about the product. As I told you earlier, everything is predefined. The quality of the product is predefined one. Now, moving on to the next one, it is the product design. Based on this profile, I will design my product because of the sound knowledge of that particular product. And next comes the process. If I know the product, I definitely can show what my process is. Because of that, again, the sound knowledge of process understanding. Now, a person who knows about this product and process, he definitely can control the strategy. The product as well as the process design, definitely know about the control strategy. If he knows how to control, how to minimize the errors, further it can be improved. The process can be improved. The actual yield, the manufacturing efficiency will be increased by this means. If somebody is not understanding, please let me know if you have any questions in between, because I'm just rushing up. Next slide, please. Now, as we have spoken, what is a QBD element? The first thing is QTPP, which I have spoken just now, quality target product profile. That is nothing but my product profile. It's a prospective summary of the quality characteristic of a drug product that ideally will be achieved to ensure the desired quality, taking into the safety and efficacy of the drug product. Okay, so this is nothing but we call this as a specification. Now, in that specification, what is that thing which actually is critical quality attribute? QBD is to find out what is the critical attribute, and we will work on that critical attribute so that you can control the failures, you can control that errors. You will not miss out. It can be a physical, chemical, biological, or a microbiological property or characteristic that should be within an appropriate limit. It should be an appropriate limit. For example, if it is a assay, it should be 90 to 110, or if it is 95 to 115. It can be a range or distribution to ensure the desired product quality. These are all the two elements. Next. Next slide, please. So, now what is QTPP? It's a set of elements which actually defines the drug product. It can be an appearance, it can be a like impurities, it can be a say, It can be a water content, anything which actually contributes to your product specification. And how much you want it, how much you want to achieve, also it is well defined. The target or goal which is set in advance. It's all about the product and process knowledge. It's a guide. You can say it is a guide to your product development. So it is important to understand what is a quality target product profile is. And when it has to be defined, of course, at the start of every development. At the start of any development, you have to define this. Now here comes your quality target product profile. So your quality attribute, what we discuss over here is the dosage form. Okay. So what is our target? What is the criticality over here? This we need to see it. Okay. So dosage form, potency, pharmacokinetics, appearance, identity, assay, impurities, water, content uniformity, resistance, dissolution, disintegration, microbiology, all these elements constituted drug product. These are all the elements of a drug product specification. This is in fact a drug product specification, and we need to fix the target based on the sound knowledge, whether it has to be a tablet, whether it has to be a capsule, or whatsoever it is, the dosage form and the way it has to be done over here. It has to be defined all through prior knowledge. Potency, how much active pharmaceutical ingredient should be there in each product, that is also defined over here. Now, you have developed a product. When it goes to the market, and when some patients, if they take it, when it will be released in your body, that is kinetics, and whether it is an immediate release dosage form or sustained release dosage form, everything will be predefined, and the Tmax also will be defined here. And appearance, tablet conforming to description, shape and size, everything is well defined. That also need to be well defined. Even the appearance, some omit this appearance, but it is very well important while designing a product. Without that, we will not move through. The first test which QC do is the appearance. If appearance fails right immediately, that time the product will be rejected if the appearance is not good. The identity, again, it is for positive. Assay, limits, impurities, everything. So out of all these targets, whatsoever it is given over here, you will not believe this appearance is a critical parameter. This identity is a critical parameter. Assay is a critical parameter. Impurities is a critical parameter. Dissolution is a critical parameter. Content uniformity also a critical parameter. And rest of the things, whatever we say, like disintegration is not a critical. Why we are saying disintegration is not critical? Because the dissolution is already a critical one, which we have already defined. So we are saying it is not critical disintegration. Microbiology is very much critical. As I told earlier, rather than manufacturing a product, it is very much challenging the environment, to control the environment part. So for example, in case of a class 100, that is a sterile product room. If you want to build a sterile product room, so to enter into the class 100, you have to pass through the changeover. You have to pass through the class 1 lakh, class 10,000, class 1,000, then only you can able to reach the class 100 facility. So you have to pass through four different types of room. That's how it will be designed. Now, QTTP and CQS. This QTP forms what we have seen it. Now, how this contributes as a critical quality attribute. So assay, it defines the efficacy, how much percentage is available, and impurity, it belongs to safety, CU, content uniformity is efficacy, and dissolution is the efficacy. It's all these parameter efficacy. Why I'm saying this safety, impurities. Impurities, one such product is ranitidine tablets. Do you know why? There is an impurity called NMDA, which is actually found out as a potential impurity, which is identified as a mutagenic impurity. Mutagenic impurity in the sense which alters your DNA characteristics. Nowadays, whoever is manufacturing such kind of medicines, they are withdrawing all the products and they are re-establishing the limits. And they have to, it has been told to all the manufacturers, whoever is having NMDA, NDMA impurities in their specification, they have to reanalyze all those products and the report has to be submitted to FDA within a year. That's how the potential of the presence of an impurities in a product, which affects the patient safety. Next slide. We have seen the QGPP and the critical parameters, quality attributes. Now comes the material attribute, how a material will contribute in terms of developing a product, which can be again a physical, chemical or biological origin. It's an input material that should be within the appropriate limit. Again, it can be a drug substance or excipient. You may be using the API, you may be using any solvent, even you may be using a water into the system. When you develop all these things, of course, you have to take care of water because water plays a major role, major contribution, because due to the microbiological growth, because of the purity, it contributes a lot. Now comes to the process parameter. A process parameter, definitely it is having an impact on the critical quality attribute. Process parameter, obviously it results from the equipments. Next slide, please. To understand all these concepts, I have just put this slide over here just to understand the process flow. When you manufacture a drug product, say for example a tablet, you should define what is a material, what is the particular process you are going to adapt, what are all the controls you should have, for example in process controls, and what is that critical quality attribute you are going to define it over here. For example, if it is a material, the API material whichever you use, an MCC, microcrystalline cellulose, magnesium stearate, lactose, this will all disintegrant type of excipients, which you do it during blending. It has to blend with the API. That is the first step of any product development, any tablet manufacturing. Okay. Now the in-process control. What control we should have it? If when it is in blending, it should blend uniformly. So the blend uniformity is the important factor in terms of manufacturing a tablet. First is dose uniformity. Now comes the granulation, you do it. Density is the major factor. Why? Again, in dissolution, it may or may not release it. Milling, lubrication, compressing, and packaging. These are all the different types where other input materials will also be added. So it is very much essential in a formulation industry. In-process control check is very much essential and it is seen as a very serious steps. If it fails in the in-process controls, it will not be taken to the quality control for further analysis and the product will not get analyzed. This compressed tablets only will define the assayed uniformity and the resolution appearance. This is about the process flow. We'll understand the next step in the next slides. Next slide, please. Now we have seen the process flow. We have seen what is quality attributes. We have to define now how we are going to implement this QBD, that is quality by design, how we are going to control your process. Just to make you understand, I have just taken only one step which we have defined right now, blend uniformity. One step, this is just a one step in how much amount of analysis you have to do, how much amount of work you have to do, just look into it. This is one step. Likewise, if your process contains 7 to 8 stages, this many analysis need to be done, this many need to be product experiment need to be done before even proceeding to the development stage. So blend uniformity, of course, what will contribute in the blend LOD that you need to see, like APA or diluent, which constitute the blend LOD. Next comes your particle size. Again, there should be a brainstorming session. Why, how, your API. particle size will impact the product uniformity, blend uniformity. Again, you need to see what is that one thing which contributes your method validation or sampling or method, how it should be in an analytical perspective. Particle size, how it will affect lactose, other excipients, everything you need to see it. When it comes to manufacturing, each and every room in the manufacturing area has to be controlled by temperature and the RH. Why? If your product is a light sensitive material, the room has to be designed in such a way that there should be a dark place so that your material will not get degraded. Same way, if it is very much moisture absorbing substance, the relative humidity of that particular room has to be controlled. So wherever you are seeing that red colors, it indicates there is a potential high risk if you don't control that particular parameter. Rest green, of course, there is a low risk, but APA, MCC, whatsoever relative humidity, I'm talking about number of revolutions when it comes to the equipment part. If it is not maintained properly, if it is not calibrated properly, it will not give the accurate measurements. So it is essential to consider the calibration of the equipments too. Every time, preventive maintenance time, like how long you're going to do, 1 week, 2 week, or batch to batch, or product to product, how you're going to clean the equipment, how you're going to maintain the equipment, when you're going to calibrate the equipment, it also should appear in the documents. Now comes the process parameters. You have loaded all these materials in your machine, and which blender you will use it, in which order you will add the excipients or the APA, or when will you load, number of revolutions. If it is not functioning properly, out, your product will be rejected. Likewise, this is one stage you have to do all these exercises. If you move on to the next one, drying. The drying stage also will be having these many number of parameters to be experimented. If you see here, the input material, we have identified all those things like blend uniformity. We have seen is why this granule surface area is important, ribbon density, compactability, APA, PSD, particle size. Particle size will have a major impact in dissolution. We have given this input into the process when it comes to compression. Now, your critical process parameters are your pre-compression force of an equipment in which you have loaded, the compression force, compression force, press speed, die fill, where you will where your tablet will get punched, and feeder, ejection, height of the drop, everything will be measured. Even the process geometry, tooling geometry. When the inputs come into the process, your output will be your tablet. Whatever you're defining as your quality attributes, appearance, tablet, weight uniformity, content, hardness, friability, everything comes out well. Of course, your yield and production rate also will be in a higher state. Hope by this time you would have understand why this QBD has been introduced. First, you are designing your specification, product profile, and you are going to do, like, I'm going to define this specification. My target is this, and I'm going to achieve. To achieve this output, you have to reverse the process. From here, what are all the inputs you have to do? What are the process you need to do? Reverse work, of course. You're defining it, well-defined process. If you do all these things, your output will also be an excellent one. Next slide, please. Using risk assessment. We have seen all these critical material attributes. We have seen the process parameter, which will affect the drug product sequence. We have seen it. Now, how do we will come to know about this is a risk factor or this is not a risk factor? Next one, please. Everything is a calculation. Risk assessment, it has to be done in one simple way. There are 3 types: low risk, medium risk, and the high risk. whatever has been indicated in green color, which is called, it's a known risk, but further investigation is not required. A smaller amount that is very low, it can be negligible. And whichever you are seeing in the yellow color, it is acceptable, but investigation is required. Whether if it increases, there may be any chance of risk. Risk in the sense product failure. That is called risk here. And the red one, it is unacceptable. If you change anything, if you change the die of a machine, compression machine, definitely you will not get your desired product. That is for sure. That is risk. Your product will definitely fail. That's how the risk assessment should be done. Next slide, please. Now everything cannot be done, like each and everything. If you go and search, research, it will take a lot of times. So there is a tool used to measure all these QBD elements, that is called design of experiment. Whatever the process parameters, what are all the analytical parameters, what are all the equipment parameters, everything will put together. You can put it in a software called design of experiment software. Okay, it actually uses a basic statistics, mean, median, mode, normal distribution. This is all standard deviation. Everything is a basic statistics. There is something called factorial design. 2 factorial, 3 factorial, multifactorial. Are you going to do one change at a time, or are you going to do 2 changes at a time? It will design the factorial design. 2 into 2. For example, if you're going to do 2 equipment modification and 2 process parameters, it is called as 2 into 2 factorial design. The result will be given based on you need to select your material. You need to select your equipment, and that particular change only you have to do at a time. So likewise, you can list out how many trials you want to do it, that many you will be doing it, and you will get a desired results. So this is in terms called as tools, which is used to measure the QBD element. Next slide, please. Now, we have designed our product, the desired output. We have plenty of knowledge about the product, no doubt in that. We have a control strategy, as we have decided in the last few slides. All the specifications, whether it is 95 to 100, 90 to 100, or 80 to 100, whatsoever it is, we have already defined it in a very controlled manner. Now you have to design some space. Your control space should be very stringent so that it will not go out of the limit. If you control your process, the product will not go out of this design at all. So that's how this has been designed. See, I have put it in a very simple manner so that you will understand. Knowledge is not just enough. The control space should be in your design space, not in your knowledge. In knowledge, if you put it in the knowledge, again the trials, you will be taking only the trials. It should be in a design space where you have defined your pre-mentioned attributes. Next slide, please. So we have seen what is QBD, what are all the quality target profiles, what are all the quality attributes, how you have to measure those attributes, how you have to do risk assessment, how you have to put it into the process so that you can develop a strategy. Formulation strategy, we have mentioned it. We have mentioned the analytical strategy too. So the basic idea is your formulation process, design and understanding is very much essential. And the material, whichever you are going to feed it here, you are going to do risk assessment. You're going to define what is your critical quality attribute, what is critical in your process, what is critical in your material. This need to be defined. Identifying these 3 elements contributes the product quality. And the ranges also has to be fixed. Like, for example, critical material attribute, CPP, limits for critical quality attribute, say how much it should be. Your API, What is that particle size? It has to be defined. What is the dissolution range? Not more than 80%, not less than 80%, something like that. This is how you build the control strategy. Now you have built the control strategy also. Next slide, please. Well, a product is designed in a well-defined, well-designed facility by means of a well-designed equipment, by designed, well-defined process, and you're going to do the total product development. You're going to start, you are analyzing everything in brief, you are identifying the key requirements, you're into research and which type of equipment material you need to select, you're generating those ideas, you're putting these ideas into development. Then this will go for a validation. After the process validation, this will go to the production for commercial manufacturing. Testing will be done in the quality control, the yield, how much you are going to get it. The product implementation will be over in that step itself. Again, you are not going to evaluate it in terms of stability study. So once you have got it, doesn't mean that your product is consistent. No. You have to analyze this product by time to time. Next comes your redesign. If anything failures happens out of specification, if it goes out of the trend, again, it has to brought back to the production department and reprocessing has to be done. That is called redesign. Until you reach your level of confidence, your level of designing, this cycle has to go. This is the overall design process. Next slide, please. All these days, we have heard, we have learned, these are all the 5 pillars: production, process, facility and laboratory, system and packaging and labeling. But the trend has been changed. Industry has changed the trend. Now this is not only the scope of the pharmaceutical quality systems. Next slide, next enter. One more enter, please. The entire prospect of the quality system. Prior slide. Previous slide, please. The entire quality system has changed from this part to this part. This is a new quality paradigm. If you go and visit an industry, you see only these things: product profile, CQAs, risk assessment, design space. Everybody will be talking about this, identifying the QRM, design space, control strategy. This is about a new quality paradigm which industry is following now. Due to this, the product which is getting launched after 2 years, now the product life time has been reduced to 1 year. This is the traditional method which is on the left side. This is the new quality system which has been present in the present pharmaceutical industry. This is not easy task, like to bring from this change from the traditional way to the new quality system. So what is the one it factor which made possible to bring out the traditional system into the new quality system? How we can build, how we can design the quality of a product from the initial itself. How is it that possible? Next slide. It is not just about selecting an API, not just selecting a raw material, not just selecting an equipment. The entire culture in the organization has to shift from the traditional way of approaching a development process to a very new process. Okay, that is called quality culture. If you go and visit a pharmaceutical industry, you will understand. It starts from the training. It starts from the knowledge. It's from the awareness, intent, capability. It is the responsibility of the management to ensure there is a proper leadership skills which has been exhibited by each and every scientist. Whether there is a document quality is available, staff staffing levels, regulatory focus is there or not. All these built together will bring out that quality system which we and of course the pharmaceutical organization expects. Without this, of course, earlier it was told like there will be always challenge between a formulation scientist. Like I developed this product, They name it that I developed this and that. But nowadays, that is not the case. QBD is not an individual work. QBD is a teamwork. So whatever has been put forth over here, leadership, IT, tools, staffing, quality system, training. Of course, training is a vast area where the people, those who are eligible, even those who are entering into the analytical development, they have to qualify them as an analyst. Whatever, if they are going to use 18 techniques, they have to qualify in the techniques, then only they will be allowed into the work. This is the same case for the formulation scientists too. They have to demonstrate their competency. Otherwise, they will not be allowed in the research and development facilities. So altogether, this quality culture, which shifts from the tradition about by design, you can develop a product as you want. Thank you. That is the end of the presentation. Next slide, ma'am, so you can show it to them. So I have just given a glimpse of this QBD, quality by design. It is not about the final product. It is about the starting material, which should be of quality. So the entire approach is to make you understand how the industry has changed. So you can ask any questions or any doubts if you have. I'm ready to clarify that to you. Hello, am I audible? Yeah, I'll wait for your questions. I know this is a very new topic and of course may not able to grasp everything in a one single forum. There is a lot to discuss to begin with. Thank you. And somebody has asked me about, elaborate about microbiology. Can you please unmute yourself and ask so that I can understand the question? Deepali.
Yes, ma'am. Please elaborate about the quality control in microbiology.
Okay, so you're very much interested to know about microbiology. See, microbiology, see, quality control is a part of quality system, that's it. And microbiology, as I told you, it has to be separated from the manufacturing unit. It should be separated from the quality control. Even in fact, the procedures which has to be followed in the microbiology should be separated out. It starts from the water, whichever you are going to use it, the culture, the media preparation, everything. So each and every step you follow, it should be well defined as in terms of procedure. So when you go and see in an industry, you doesn't have to, research many things. everything will be available, which is a predefined thing. Okay. Starts from the media preparation, start from the which culture you have to use, how you have to analyze. And one important thing is the specification has to meet the pharmacopoeial standards of the respective regions. If you're going to, for the domestic usage, it has to meet the Indian pharmacopoeia. If you're going to use it or market it to in US, it has to meet the USP, United States Pharmacopoeia. That is the Bible of all the pharmacopoeial organization. You may be knowing it. If it is in the British side, it's the British Pharmacopoeia. As you are mentioning about quality control of microbiology, you have to stick to these pharmacopoeia. What is defined in the pharmacopoeia, you need to follow. Not less, not more. So what you understood from this concept, quality by design? Is anyone has understood the concept correctly? Just would like to know about it. Do you have any more questions in that? Thanks for the wonderful comments. Anyway.
Is there any software available to perform risk assessment?
There is no software available for the risk assessment. Risk assessment you have to do by yourself based on the inputs. Of course, for QBD it is available. That is called DOE experiment software. That is available, not for the risk assessment. There is a chart like you have to scale it 1 to 10 and X and Y axis, like as I said, low, medium and high. You have to put it in those boxes and you have to rank it. Risk assessment comes with the ranking. As low as possible, the risk is low. If the number, the scale is more, it is highly risk. That way.
That standard numbering, is there any standard given from ICH or some other organization?
Yes, it is given in the ICH. It is given in the ICH. If you are well aware about ICH guidelines, please refer 8, 9, and 10. Quality risk assessment given in the Q9. This can be used in any way, like not just in the quality process, risk assessment. And even you can use it for any of your own activities also. Hope I have answered for your questions. Hello? Oh.
Even though
Is
we
anybody
have a
talking?
little knowledge of the body, but the way how Mrs. Sudhera presented the topic made us much more aware about the topic, a systematic approach to pharmaceutical drug development. And thank you, ma'am. Thank you for the nice presentation. Now we will go ahead and take some more time for question now. Just a
Thank you so
reminder,
much.
please type your question into the question box in your control panel or raise your hand. Now I request Mrs. Tina Cakes, Professor, Kennedy College of Pharmacy, to propose vote of thanks. Thank you, Madam.
Good evening, all. Myself, Dr. Sina KS, and I am here to propose vote of thanks for today's orientation session on unlocking innovation through quality by design, a systematic approach to pharmaceutical drug development organized by Department of Pharmaceutical Chemistry and IIC of KMB College of Pharmacy. First of all, I would like to express our sincere thanks to the speaker, Ms. Udaya Raj, for giving an excellent coverage on the topic, quality by design. Thank you, madam, for making this session interesting and meaningful. We are grateful to our management for their support and motivation. I would like to thank our principal, Dr. J. Nandakumar, for his worthy guidance, and I also extend my thanks to our professor, Dr. Harish Babu sir, my dear colleagues, for their great support. I thank our system administrator, Mr. Eldos and Mr. Sibi, for the technical assistance. I also express my humble thanks to all the faculties, research scholars, and students from various organizations for the time and attention. I will end my speech here. Once again, thank you all.
Thank you so much. And it's a wonderful experience with you all. And thanks to Dr. Nandakumar for giving me this opportunity. Thanks all.
Thank you, ma'am.
Thank you so much.